Hello everyone, welcome to Assist Biology. In this video, we will discuss anchor cell formation in C. elegans, and this is from the unit of developmental biology. I am Ashish Kumar Das, student of M.Sc Animal Biology, University of Hyderabad. And this video will be helpful for BSc and M.Sc student, CSIR NET, ICAR NET, DBT JRF, ICMR JRF, GET Lab Science student. And if you are new to this channel, please subscribe the channel and press the bell icon for getting more updates about the new videos. So what we will learn today? First of all, we will see what is anchor cell. Then we will see what are the cells that are involved in the anchor cell formation. Then we will see what are the signaling molecules involved in the anchor cell formation. Then we will see how the process of signaling will take place. Then and many more. So in our previous video, we have discussed the vulva formation in C. elegans, and there we have seen what is the role of anchor cell. Let us recapitulate on that. So first of all, the position and the function of anchor cell. So in this diagram, we have seen in our before slides or before videos, we have seen the gonads, the anchor cell and the VPC, that is the vulval precursor cell. So the anchor cell releases the lin-3 protein, that is the signaling molecule, and this will lead to the induction of the vulval precursor cell, and they will become the vulva. So the anchor cell acts as the cell that sends the signal or that secrete the signaling molecule, that is the lin-3 protein. So the position is it is present below the gonads. This is evident from the diagram, the gonads and the anchor cell, and it secretes lin-3 protein, that is a epidermal growth factor that induces the vulval precursor cell to become vulva. So anchor cell formation, as you know, the vulva is present only in the hermaphrodite, that is the hermaphrodite are the female that can produce limited number of eggs. So the hermaphrodite will only have the vulva because this is a part of the female sex apparatus, and the male individual can't have the female sex apparatus. So this is present only in the hermaphrodite individual, that is the female that have ability to produce limited number of sperm. So here what happens in the wild type individual or wild type hermaphrodite, what happens, it have two cell, that is two cell having potential to form the anchor cell. The two cells are called as Z1 PPP, or they are numbered or they are named as Z1 PPP and Z4 AAA. So Z1 PPP and Z4 AAA, these are the two cells, and they constitute a equivalence group. So why they are constituting an equivalence group? Because they are having equal potential to form the anchor cell. That is both of the cells, that is the Z1 PPP and Z4 AAA, they are having equal potential to form the anchor cell. That's why they are called as equivalence group. Although depending upon the signaling molecule they are receiving, they will become two different cells, but they are having equal potential, and at a time they can only one of them will form the anchor cell. So what happens in the wild type individual, the cell which produces higher concentration of lin-12 molecule, that is with lin-12 molecule will become the uterine tissues, that is it will be the precursor for the uterine tissue, and the other cell, that is among from the two cells, that will produce the lesser quantity or no quantity of the lin-12 protein molecule, they will become the anchor cell. We will discuss this in detail. So at this stage we must remember that the cell with higher concentration of lin-12 will become the precursor for the uterine tissues, and the cell with no concentration of the lin-12 or very little or negligible concentration of the lin-12 will become the anchor cell. So now here you can see in the wild type there are two cells that we have already discussed, that is the Z1 PPP and Z4 AAA, and it's convenient to assume both of them as cell 1 and cell 2, one cell 1 and one becomes cell 2, because while we consider it as Z1 PPP and Z4 AAA, it become quite difficult, means it will not be easy to correlate. That's why let us assume cell 1 and cell 2. Okay, assume that them as cell 1 and cell 2. So by what happens, by chance event what happens, so one of the cell produces high quantity of the LAC2 molecules. So first of all, one of the cell produces the LAC2 molecules, and the LAC2 molecules act as ligand. So this ligand, that is the LAC2 molecule, induces another cell, that is the cell 2, here you can see the another cell, cell 2, to produce lin-12. So as a result, by the induction of the cell 2 by the cell 1, that is the cell 1 is secreting the ligand, that is LAC2 protein. So the cell 2 will now produce lin-12 molecule, and the lin-12 molecule level increases in cell 2, and then they will form the uterine tissue. As you have already discussed, the cell with higher concentration of the lin-12 will become uterine tissue, that is the precursor for uterine tissue. So here the cell 2 will become uterine tissue. So the whole process is a by chance event, because you don't know which cell will become the the anchor cell, and which cell will become the uterine tissue. So this is a by chance event. One of the cell by chance will produce high quantity. This can be cell 1 or this can be cell 2 also. So here we are assuming cell 1 produces high quantity of the LAC2 molecule, that is the ligand, and this stimulate other cell, that is the cell 2, to produce a lint-12 molecule. Here you can this is quite easy to understand in such a way that if a cell is producing LAC2 molecule, ligand molecule, that it will be unable to produce lint-12 molecule again. That's why you can say this is a kind of request to cell 2, that the cell 1 will request cell 2 to produce the lint-12 molecule, and the cell 2, as the concentration of the lint-12 molecule increases in the cell 2, they will become the precursor for the uterine tissue. So what are the steps? As you have already grammatically discussed this, we have discussed this in the form of a diagram. Now we will see the steps. So first of all, it involves a delta notch pathway. So delta is the ligand here and the notch is the receptor. So ligand receptor pathway is also called as delta notch pathway, and this is a kind of juxtacrine signaling. So you have seen different kind of cell signaling, that is the autocrine, paracrine, juxtacrine, endocrine. So juxtacrine signaling is a mode that is contact dependent signaling. So what happens? The LAC2 molecule is the ligand and the lint-12 is the receptor. So this is a by chance event. One cell that will secrete the LAC2 and induce the other cell to produce lint-12, and higher concentration of lint-12 will become the precursor for uterine tissues, and cell with no concentration, less concentration, will be the anchor cell. We have already discussed this in the diagram, you can see here. So no concentration will be the higher concentration of lint-12, the concentration of lint-12 is increasing, that's why they will become the uterine tissues, and the cell 1 is having the LAC2 molecule and it is without the lint-12 molecule. So the level of lint-12 is low here, so it will become the anchor cell. So the whole process can be summarized in these points. You have to remember these points. This is the end of the video. Thank you for watching.